So today I will be talking about the sponges. The sponges belong to a phylum. In last class we had understood what a phylum is. Phylum is a type of ranking. I will continue with your classification in the future classes, but now today I will be talking about phylum porifera. So phylum poriferans are nothing but sponges. So with that, let me start presenting my screen. So poriferans, the word porifera means, are you able to see my slide? Unmute kore.
Yes, sir.
Huh? You're not able to see?
Yes, sir, I am able to see it.
Okay.
Yes, sir,
Okay.
able.
Okay, fine. Al Shifa, you are able to see?
Yes, sir.
Okay,
Sir, I am
fine.
able to see.
Okay, you are able to. So porifera means Pori. It means pores. Pores are nothing but hole. So if they have a body which looks something like this, they have lot of pores throughout the body.
So, ostia.
First, right now, ostia is a later. Yeah, they are ostia, but consider them to be pores. They have lot of pores over their body. These pores are known as ostia, of course. They have pores all over their body. Therefore, they are known as porifera. So these sponges, they include around, this is not exact figure or correct figure, around 5,000 species, almost all of which are marine. There are about 150 freshwater species, member of the family Spongilidae. So a group known as Spongilla, they are the only freshwater sponges. Spongilla. They are the only freshwater sponges, and the rest are all found in the sea or ocean, marine. Freshwater means in rivers or ponds or lakes, and marine means in the sea. This occur worldwide. Sponges, they occur worldwide at all latitudes from the intertidal zone to the deep sea. So what is the intertidal zone? Says for example, this is the land, and from here this is the sea. So this region is this is the sea, and this is the land. This region is the land. So up to this region, from here to here, this is the intertidal region, IT, and inside is the deep sea. So sponges occur throughout the world. They range from few millimeter up to 2 meters, their size. Porifera, as I told, they mean pore bearing, and they refer to the numerous pores and channels that permeates a sponge's body. So there are a lot of fine channels which are permeating inside the body of the sponge. So this is a picture of a sponge. Can you see the picture of this is known as the yellow tube sponge.
No, sir, I can't see.
কেন একটু ওয়েট করো আলশিফা ক্যান ইউ সি
No, sir,
Yes.
I can't see the screen, sir.
আমার নেটওয়ার্ক প্রীতদীপ নেটওয়ার্কটা খারাপ আছে you see you will find it in the recording, okay? So because it is sharing right now. So this is essentially, this is how the yellow tube sponge looks like. This is known this is another sponge. It is known as the barrel sponge. It's a picture of a sponge. Pritadeep, can you see this?
Yes, sir. I can see this.
But you cannot see this.
Yes, sir. Now I can see this.
Now you can see this, fine.
Screen
এটা হচ্ছে
paper দেখা-
this is the yellow sponge and this is the barrel sponge. Sponges are the simplest multicellular organism, but they lack germ layers of more complex metazoans. So what do you mean by germ layer? As you have seen in the last, the coelomate, the outer layer, you have heard that outer layer is ectoderm. Second layer is mesoderm. In between mesoderm you have a space known as coelom, and you have an inner layer known as endoderm. So ectoderm, you have a mesoderm and an endoderm. So 3 layers in the body. So that is known as germ layers. Germ layers are nothing but ecto, meso and endo, EME. In between the mesoderm there is a space, fluid filled space known as the coelom. So sponges are the simplest multicellular, they have got many cells, multicellular organisms, but they lack the germ layers of more complex metazoans. We are more complex metazoans. They have a cellular level of organization, lacking tissues and organ. I have also last class I have explained what is organization, level of organization. First is when you are talking about individual cells, they are cellular. Then cells of the same type in an organ is known as tissues. So they don't have any organs, that's what they meant to say. So they have no organs and no tissues as such. They have individual cells. Tissues like liver tissues, skin tissues, they are all tissues. They don't have that. So body is a mass of cells embedded in a gelatinous matrix. So there is a lot of cells and they are embedded on top of a gelatinous matrix called mesohyl. I will explain what mesohyl is, which is supported by a framework of spicules as well as collagen and spongin fibers. So by this I mean, these are the spicules in the body of a sponge. These are glass sponge, these are spicules, different types of spicules. These are made up of siliceous spicules. Then these are also different types of spicules. So essentially the sponge body, if I draw the sponge, and this is the body of the sponge, you have an outer layer and an inner layer, and in between you have this layer. This layer is the gelatinous matrix known as mesohyl. I have to buy a graphic tablet, otherwise writing with my mouse is very difficult. So the outer layer is known as pinacoderm, and the inner layer is known as choanoderm. I will come to that later. And this is supported from the gelatinous matrix. There are these spicules present, these type of spicules present, or they may have spongin fibers. The spicules can be calcareous, made up of calcium, or they can be siliceous, made up of silicon. So the spicules, they form the stiffening skeletal structure of the sponge and may be made up of calcium carbonate, silica or spongin fibers. So now let us look at in details on a sponge structure of a sponge body wall. So this is a picture of a freshwater sponge known as spongilla. This is spongilla. So whenever you are watching the class on your mobile, keep it in a horizontal manner, not a vertical straight manner. Keep it slanted. कात करे रखो, साइड करके रखो, सीधा मत रखो. So spongilla is the only freshwater sponge. So if you this is the sponge, you cut a section, you see the body wall. So if you see the structure of a sponge body wall, you have an outer layer here. This cell, this is the pinacocyte, and the inner cell, they are having a collar like structure, this collar like structure, hence they are also known as collar cells. They have also this flagella and they are known as choanocytes. Then this is the pore or numerous holes on their body, and they are guarded by cells known as porocytes. Water flows through this. So this is another porocyte here. Apart from that, this middle region, this is the mesohyl, gelatinous connective mesohyl, and they have these structures known as spicules. Spicules are made by cells known as, like the cells like this cell, it is known as sclerocytes. Then you have oocytes, lopocytes, different thing, and inside the sponge is a space known as spongocoel. So likewise, if you look, this is the basic structure of sponge. So sponges are sessile, that is they don't move. एक जगह थाके, एक ही जगह पे रहता है, ये यहाँ हुआ नहीं जाता है. So एक जगह थाके, they don't move, they are sessile and depend on water movements to bring in food and oxygen and remove waste. So water enters the sponge through the pores, outside pores, and this water is responsible to bring in food and oxygen and remove carbon dioxide and other nitrogenous wastes. So water enters through in a sponge. It and on the top there is a hole. So it is known as the osculum.
But sir,
Ah.
what's the function of the flagellas on the body wall?
আসছি সব আসছি. So sponges are sessile, they don't move, and there is a water which enters and it brings in food and oxygen and removes wastes. Sponges, they generate their own flow of water having a unique water current system. And this water current system, so the water has to enter inside the sponge. So if you look at the sponge, a sponge, this is the sponge. If you look at the sponge, the water enters through this ostia or ostium or through this pore. It goes into the space inside, that is known as the spongocoel, and it exists outside through an aperture known as the osculum. And also, you know, this is the interior side or inside the body, and this is the exterior side outside the body in the water. Also, you know that the inner layer have got choanocyte cells and outer layer pinacocyte cells. This is this cell with a collar is known as choanocytes. Collar and a cilia, a flagella. It is a flagella, this whip-like structure. This is the flagella whip. So, what is the function of this flagella? This flagella beats. This flagella beats. That is, it moves, and this movement, it generates a force, and that force, it draws the water inside, like what Pritodip was saying. What is the function of this flagella? The function of this flagella is that while it is beating, it is pulling water from outside into the cell. So when water comes inside this, it brings in oxygen, and all carbon dioxide which is formed, it is taken out, and food particles, small zooplanktons, phytoplanktons, food particles, they come, they are trapped by this cell, and this cell ingests this food particles and gets nutrition. But you don't have an organ system, so it is like individual cells getting their nutrition. So sponges generate their own flow of water, having a unique water current system. So water enters through many small pores called ostia and exits through fewer larger oscula. So if you draw the sponge, on the top, of course, you have this is the osculum, and you have pores all over the body known as ostia. Through these pores, water enters. So these huge circular structures you see inside, this is the oscula. Underwater it is very colorful. If you pour some light on top of, you can see beautiful colorful structures. So this is a sponge. Picture of another sponge. So these ostia or these openings are connected by a series of canals which are lined by choanocytes or the flagellated collar cells that maintain current and filter out food particles. So these choanocytes, they beat, they maintain the water current, or water is pulled inside, and there are a lot of series of canals. A picture of a canal, I am showing you the picture of a canal. This is the picture. So these are the collar like cells. Food particles are trapped inside these collar when the water enters, and they are ingested inside these cells. And beating of this flagella, it pulls the water inside. I am just showed a individual cells. Now I am showing the canal, how the canal looks like. So this is like this. Outside, this is an ostia here. This is the ostia here. Water enters through this. This outer layer is the pinacoderm, or which contains pinacocytes. This red line here inside, it is the choanocytes, or the flagellated cells are located in this region. And This beat and pull the water inside. Water goes inside osculum and exits outside through the water enters through the ostium into the spongiocele and enters out through the osculum. This is the osculum, this region here. This is the osculum. So let us go back to the former slide.
Sir, ostium is the singular form of ostia.
Yes. So the choanocyte collar consists of microvilli joined together by delicate microfibrils which filter out tiny food particles. So if you have a look at this picture, just hold on, it's coming. If you look at this picture here, this is a choanocyte cell. It has got, this is the collar. They have got small microvilli over here. These small, these lines which you see, this small bars, they are this is the microvilli. There it's enlarged, and they have got cross microfibrils. So when the water comes, the water is entering and bringing in food particles. It goes inside. It is filtered out. So larger dirty particles, they are removed, and the proper food containing particles, it enters inside. Water enters inside and it exits from here. So carbon dioxide, oxygen also comes here with this water, and food particles also come inside, and carbon dioxide is also removed, and also nitrogenous wastes are also removed. So this is how the collar cell works. The feeding takes place. The beating of the flagella draws water through the collar and out the top. Particles too big to pass through the collar get trapped in the mucus and slide down the collar to the base where they are phagocytosed. So they pass through the collar, get trapped in the mucus. There is some mucus in the microvilli, and they are phagocytosed. Phagocytosed means the cell engulfs them, as you can see in this picture. A food vacuole, the food is here. The food vacuole takes the from the cell, takes the food inside. So this is phagocytosis. And this is what the picture which I showed just now. So sponges can filter enormous volumes of water, as much as 20,000 times the volume of the sponge. It can take filter 20,000 times amount the volume of the sponge in 1 day, in 24 hours. So a lot of water is filtered. Sponges mostly consume bacteria and may filter out as much as 90% of those passing through. So only 10% of them they mostly consume the bacteria. There are some carnivorous sponges. They capture small prey such as crustacean. Those who come through these small crustaceans, small like shrimps or prawns. The prawns are huge. The kada chingri thekeo choto, very small, micro, like Daphnia and all these things. They enter. They are crustaceans. The Daphnia enters into it. Cyclops, they enter and they are captured. So members of the family Cladorhizidae, they captured their prey using spicules that act like velcro to hold the prey. Velcro, do you know velcro? Catch, you open. So this is a velcro. If you can hear the sound of this, you can understand what this is. I'm opening something. This is velcro. Basically, I'm showing you. I'll put the camera on and show you what a velcro is. So, can you see me on the screen?
Yes, sir.
Can you see this object?
Yes, sir.
This is Velcro. It sticks again. This is Velcro. So let's get back to the screen. Some sponges also supplement their filter feeding by hosting symbionts such as green algae, dinoflagellates and cyanobacteria that provide nutrients to the sponge. So they do photosynthesis and provide oxygen, and they make sugars. Green algae, they make sugars by the process of photosynthesis, and they provide nutrition to the sponge. So types of cells in the porifera. Sponge cells occur scattered through a gelatinous matrix called mesohyl. I showed where the mesohyl is. Spicules are distributed through the mesohyl, as are several different specialized cell types. I have shown how the spicules are organized, but still I will have a better picture. I'm going to try to show you a better picture. So these are spicules. As you can see here, you can see spicules are organized inside the body. This side is the interior side and this is the exterior side. Excuse me. Okay. So the first type of cells are the archaeocytes. They move around within the mesohyl. So let us show where the archaeocytes are. So first let me erase all what I have drawn here. Just hold on, I am just erasing this part so that you can see this much better. Just hold on, I'm just wiping this region. So I'll increase the size. So have a look. So this is what you see over here. This cell over here, this is the archaeocyte. This big cell which you see here, this is the archaeocyte. So what is the function of archaeocyte? Archaeocytes, they move around within the mesohyl. They are amoeboid in appearance. Amoeboid means a structure irregular, something like this, like an amoeba. This is amoeboid in appearance. They are amoeboid in appearance. As you can see over here, they are amoeboid in appearance, like an amoeba. They are amoeboid in appearance. These archaeocytes, and its function
irregular shape.
Irregular. They are amoeboid and they carry out several tasks. They phagocytose particles and receive particles for digestion from choanocytes. I am now going to show you on the internet what phagocytosis looks like. What a phagocytosis looks like, a picture.
So it's the process in which WBC attacks the antigens.
Not only WBC, it happens everywhere. So this is a picture of if you can see it over here, this is the what happens during phagocytosis. A food particle is here, and these arms come out and they trap the food particle inside, and then the food is digested inside. This process is known as phagocytosis. It happens in WBCs also, it happens in many places. Even in your sponge feeding, the phagocytose means the process is like this. So let's get back to the slide. Okay, they phago and receive particles for digestion from choanocytes. They can differentiate it into other specialized type of cells. So as the requirement, if they need more choanocytes, they can differentiate and change into choanocytes. If suddenly some choanocyte layer gets damaged, they need more choanocytes. These archaeocytes, they get converted into choanocytes and they work as choanocytes. They secrete structural components. Specialized archaeocytes known as sclerocytes, spongocytes and colonocytes secrete spicules, spongin and collagen. So sclerocytes secrete spicules, spongocytes secrete spongin and colonocytes secretes collagen. So the other is the choanocyte, the collar cells which I had talked about so long. So the protruding end has a flagellum and it moves water through a mesh like collar. You have seen the collar where small particles are trapped. Then you have the porocytes. AJ, this pore or ostium is guarded by cells known as the porocyte. This is the porocyte cell here. এই জায়গাটা porocyte. Then you have the pinacocytes, this outer layer here, pinacocytes. Then we come, you have the choanocytes, you have the canal system. The canal system, in details I will be talking about it. Predominantly is the asconoid, cyconoid and leuconoid system. These systems differ from each other in increasing complexity. Asconoid is the simplest water system. So let us look at the canal system in details in this particular diagram. So let's go to the canal system, diagram of the canal system. So this is the asconoid canal system. Very simple. You have water flowing from outside through the ostia into the spongocoel, and it goes out through the osculum. Cyconoid, it's a bit more complex. This is one asconoid system. So several asconoid systems, these fold together inside to form finger-like projection like the cyconoid, and many cyconoid systems together form a leuconoid system. So leuconoid system is the most complex, and what you see here, these spaces or these canals, these are canals. So water enters from here into this finger-like structure, then from here it goes into this canal, from this canal to this canal, this canal to this canal. So this is a system of canals which is present. From here to this canal to outside, this is also a system of canals present. So water flows through a system of canals. This is what you mean. There is a large osculum and the cavity inside is known as spongocoel. So this is the same picture which you have seen. Water enters from here, goes through inside from this canal to the spongocoel, and ultimately it exits outside through the osculum. In the leuconoid you can see water enters through this, follow the blue arrow, enters from here, goes inside this, goes through this system. Here all food is captured and food particles are captured, and then oxygen carbon dioxide exchange take place, and the water comes from here, and it goes out through the osculum. This is the most common type of sponge, the leuconoid sponge. The modern sponges are Euspongia. They are all belong to this leuconoid sponge. Then you of course have the syconoid, which I explained. This is the asconoid system. Example is Leucosolenia. This is the syconoid system, finger-like projections, and this is the leuconoid system. I will explain in details, much more details, when I in the coming classes. But what you see is that you have three types of organization in sponges in the canal system. Now let us talk about reproduction in sponges. Sponges reproduce both sexually as well as asexually. They are hermaphrodites also. That is, they produce sperm and egg from one sponge. Sperms are shed into the water and taken up by other sponges. Individuals with eggs use special cells called archaeocytes to transfer sperm to the eggs, and fertilization takes place. They develop into a ciliated larva. The ciliated larva, I'll come to the picture of the ciliated larva. So they develop into a larva known as amphiblastula, and another larva known as parenchyma. So the zygote develops into this larva, larva develops, and larva develops into the sponge. Zygote, coleoblastula, it forms stereoblastula, this structure, and then ultimately the sponge. This is how the development of the sponge takes place. So let us talk about totipotency. So if you cut a sponge and take some cells of the sponge, these sponges, they will reorganize and form a new sponge. So this is known as totipotency. It is there. If you cut your fingers off, your fingers will not grow back. But if you cut a part of a sponge, the sponge will grow back. So with this, I would like to end my talk over here. Today's class on an outline of the sponges, and I will send you my note today itself. So I will send you my notes today itself. And do you have any questions regarding sponges?
No, sir.
Okay, then bye bye today.
Okay, sir. Bye. But sir, your next class on Monday, 4 p.m.
Monday or Sunday?
Monday or Sunday.
Sunday te hobe.
Okay. Okay, sir. Okay, Sunday, 4 p.m.
Yeah.
Okay,
I will
sir.
give you the time. I'll organize the time and date. Possibly I will do it on offline one day, but online I can do 2 days. When we have offline classes, it will be, perhaps I will fix a day. Once the offline classes start, it will be one day is good enough, and one day we will have online. That will be sufficient. And
But sir, that is much better than online classes.
huh?
But sir, offline classes are much better than online classes.
Yeah, I don't agree. I absolutely don't agree. Because I take a class. Yeah, you're quite new to online classes. In an offline class, I will not be able to teach it like this. Definitely, I cannot explain each of these diagrams and all these things. I just cannot do it. It is impossible. These are the modern technique of classes. So you have to get used to it. That's the thing. The sad part of our education system is that whether it be Shomnath or whether it be TKB or whoever, all our school education, they spoon feed you like you are a small baby, that you have to take a feeding bottle and put the knowledge inside your mouth. No, that is absolutely the wrong way to learn. And precisely that is the reason, because of this spoon feeding culture, that 99 of percent of people who attempt for medical, they fail. Basically, they don't have a conception at all of the subject. So to develop a conception, I would say using ICT, information communication technology, online teaching, that has been much more effective. People who have been successful with online classes, proper online classes, they are, I've seen that they are in a better position with their knowledge. I send them a video. There's a concept of, this is an IIT research, it's not my saying. This is a research from IIT. They had conducted a survey all over India, and in different universities. They found over there that the success rate of flip learning, where you give a video to the students, the student sees the video. It could be a video from YouTube, it could be a lecture or something, and e-material is given. The student then copies the e-material on their notebooks, and next day in the class we just only have a discussion. I don't keep on explaining this is this, this is that, this is not needed. So I think that is the much better with offline classes. So anyway, bye bye.